Hello, so I'm Todd Vision. I'm from University of North Carolina, Chapel Hill, and I'd like to talk to you today about reaggregating primary research outputs. Again, it's a case study, and again, it's from the area of bioinformatics. The case study here is in trying to understand the value of curation and some of the design choices that come in what I'll call reaggregating primary research outputs. In this case, information that's explicit in an article, but for human readers that in order to reuse in the way we want, it needs to be synthesized across studies. And it's a very common problem, and we've heard about it in various guises at this meeting. In particular, I want to talk about how we can maybe build up an evidence base that would guide, for example, funders who have to decide where to invest, what the scientific payoff is in curation versus other forms of innovation and scholarly communication. So the motivating metaphor is this wonderful one from Steve Pettifer about cows and hamburgers. And so imagine that the research as a whole is the cow, and it goes into this meat grinder. And at the end, you have the homogenized, indistinguishable mass of meat that makes the hamburger, which may be very edible for the human consumer, but perhaps you're interested in the cow. Or some aspect of the cow, or perhaps you're interested, in fact, in some aspect of lots of individual cows, and that would be, to stretch the analogy, trying to use data that's found in many different studies that needs to be synthesized before it can be taken advantage of. So there's kind of very crudely three strategies I'll talk about for how one might extract information that's found in lots of different studies that may be of value. And the first is exemplified by this wonderful paper from David Shonten's group from a few years back, Adventures in Semantic Publishing. This is actually a case where, after the fact, they took a paper and marked it up in a way that, for instance, the map data in the paper was geo-referenced and machine readable, the citations were typed relevant to ontologies, so you knew how the other literature was being referenced, the keywords were marked up with ontology. So it's kind of a best case from a few years ago of how one could make a traditional article machine readable and semantically interpretable. And of course, there's been a lot of innovation and experimentation in this area in authoring tools and literate programming and repositories to at the front end of publication to fix the issue of just embedding everything within a human-readable PDF so that it's no longer accessible to machines or synthetic research. Number two is curation, and we just heard NIH invests an enormous amount in data repositories, and much of that goes toward curation. This is an example from a survey of the International Society of Biocurators, so one particular discipline, of the kinds of objects that they curate. So maybe it's genes or proteins or pathways, but any data type which has a large biomedical audience is going to have some market there, some demand for curation of the literature, of information that's within articles into databases such as model organism databases. And the third strategy is text mining. And of course, text mining and curation can work hand in hand. And in fact, this image is from a paper about that. So here, the literature exists, and one can use text mining to triage the literature that one wishes to curate. One can extract entities from that literature. One can find relationships between those entities, such as genes or small molecules. One can annotate the evidence and the provenance of those statements and incorporate those into databases so that they can be used by others. So largely speaking, we have things that we can do at the front end, or kind of two different approaches at the back end. And we would like to have evidence that tells us how much work to invest in curation at the front end versus these other technologies, which are kind of, at the time of publication at least, they're cheap. So the project I want to talk about this in practice is Phenoscape. So it's an attempt to go into a scholarly literature which doesn't have this kind of high attention from NIH. It's like many scholarly literatures, it's maybe a little more introverted and a little more specialized. So this is sort of biodiversity phenotype data. We may be able to find relations between things that are changing in evolutionary history and the genes that are being characterized in model organisms like mouse and zebrafish and fly that can tell us that some genes may have been important in evolutionary history. Again, the ultimate aim is not biomedical, but there is, in fact, a scholarly community that publishes traditional papers. These phenotype data have been published in this form for decades and decades. And since we don't have a world in which everyone publishes structured, semantically marked up literature, it will take a very long time before we could ever do this unless we were willing to invest in curation or text mining with both legacy literature and the stuff that's being published today in traditional journals. So we don't really have a choice in the matter. We have to do one or the other, or some combination of the two. Just to give you a picture of what things look like, this is a phenotype quote from the evolutionary literature, so it can get very, very specialized. And so our challenge is to take a description like this, which I'm not even going to try to read, and to capture that in a machine-readable form, and what level of detail is necessary to make that usable in machine-readable form. So I'm going to go very quickly over this image, which just shows a simple phenotype, both for genes at the bottom in the front gray box, for a taxon, so the evolutionary phenotypes we're interested in the back box, and the relationship to ontologies above them. So the level of detail can be broken down on whether we're just trying to extract the entity, which is on the far right, which in our case comes from Uberon ontology, or whether we're trying to capture attributes of that entity. This is the shape that's affected or the size that's affected and how much detail we want to use. So simply put, it's either curation at the entity level or curation more granular than that is what we're going to be able to compare. And we want to ask, are we able to match genes from naturally occurring biodiverse taxa with genes from model organisms and human diseases in a biologically meaningful way? And our test case will be this thing that everyone learns about in their college biology class, if you've had it, which is the transition from fins to limbs as tetrapods invaded land. And so there's really nice fossil data that tells us what some of these skeletal systems look like during that transition. And we have phenotypes curated for those fossils. And from the work of Kellen Mastick, we have hundreds of articles that have been curated so that we have the expert knowledge of what the candidate genes that have been proposed by developmental biologists are. So we kind of have the knowledge in the literature that we'd like to be able to recreate, just knowing the fossil phenotypes, just knowing the model organism phenotype. So can we generate hypotheses that are realistic and useful? That's our test case. And so that the happy consequence is that overall we can. So I want to compare low granularity annotations of curation with higher granularity or finer. So low granularity is just extracting the entities. There's no detailed description like you saw in that quote. The blue is the distribution of about 15,000 genes that aren't identified as candidates by experts in the literature. The orange are the candidates that have been identified by experts in the literature. And on the x-axis is the similarity that we can see in the semantic annotation. So we are getting some separation, and that's good. But even better, if we annotate, we curate at a higher level of granularity, entities and attributes, we get even better separation between those. So it does appear that if we were to use this system, generate hypotheses to then go into the lab to test, we would actually get useful things out of it. So what do I take away from this experiment? And hopefully you do too, although this is very rushed. Aggregated scientific observations could be used to generate hypotheses that transcend the knowledge from any individual study. So we're talking here about hundreds of studies. For biodiversity phenotypes, text mining of the entities alone would have been adequate. That it gives us some separation, but there's value from human curation beyond what text mining can do today. We can't capture all those relationships with text mining alone. That requires human expertise. And human curation is going to be necessary as long as this kind of valuable information is only reported in traditional literature, and that's going to be the case for years to come. So given current funder biases against curation, which many here are familiar with, we need more of an evidence basis on the scientific contributions of curation like this. And so very happy that the results turned out the way they did. Thanks.